Okay, so it's my great pleasure to have my good friend Caroline here, although I did not invite her because she's my friend. Caroline is a professor in EECS and Data Systems and Society at MIT. She's also the co-director of the Eric and Wendy Schmidt Center at the Broad, where she is a core member. And so Caroline and I have never overlapped, although we've asynchronously been here at Cal, as she did her PhD here in statistics some years ago, and somehow I end up always running into Caroline in Switzerland and have dinner with her in Zurich, even though I'm only there at most a few days a year. It's quite a pattern. So anyhow, Caroline's work touches on quite a fantastic array of disciplines, including machine learning, statistics, biology, economics. And some of the work that she's particularly well known for includes her work on privacy-preserving data sharing in GWAS from some years ago. And one of my favorites more recently is showing that autoencoders implement an associative memory. And she's done a tremendous amount of foundational work in causal reasoning, which I believe will be the topic of today's talk. So I look forward to that.
Thank you very much, Jennifer. And thanks for having me. I'm excited to be here, back in Berkeley after doing my PhD here. Yeah, it's really nice to be back, in particular in the winter when it's kind of cold in Boston. So I will talk a little bit about causality and in particular how it relates to representation learning, and then also how it relates to experimental design. I'm not sure if is it on? It's okay? You guys hear me? Yeah, it's good? Okay, sounds good. Okay, so let's get started with why representation learning and why causality, and in particular why that is critical for biological questions. So in the biomedical field, we have many, many different data modalities, and we have, data sets coming from, or where you can image different proteins, where you can image different cells, where you can get sequencing information. You can get sequencing information at, a person's level, but also at the cellular level. And so you have all this information at many different scales, and so of course in order to have or be able to get out the most information, you may want to somehow get everything together into one joint representation, and that's definitely where representation learning comes in. But how is this different than your standard representation learning of say, when you have images from the internet and then maybe figure captions from the internet? There also you have many different types of modalities that you may want to integrate. And what we'll talk about a lot is that in the biological field, what we really want to get at is actually two mechanisms. But before I get there, just to tell you about the types of and how large these data sets are becoming in biology, and why I think it's so exciting to be working in this field, this is just for one particular platform or one lab at the Broad. This is the genomics platform at the Broad. How much data was produced, just, say here, we have the data until 2022 right now. So this is 80 petabytes of sequencing data. So this is not even looking at imagings, right? Images that generally are much larger in size. So just sequencing data, last year, how much new data was produced is 80 petabytes of data. So if you look at the curve for Twitter, it looks actually very similar. Okay, so Twitter last year also produced about 80 petabytes of data. And this is just one lab, right? One lab at the Broad and only just sequencing information. And in fact, the curve actually looks also very similar to Twitter. And if you compare that to the whole Netflix movie corpus, that's just 60 petabytes of data. So that's pretty amazing in terms of, if you're thinking about where some of the largest data sets are coming from, it is now in the biomedical field. But at the same time, although, we have this huge explosion of data, and this is just one modality, as I said, there are all these other modalities. If you're thinking about what are the key drivers of machine learning today, it's still not biomedical sciences. It is still application areas like, online advertising and recommender systems and maybe robotics, self-driving cars, like computer vision types of applications. And that's what leads to all the foundations of machine learning, and of course has been super successful and for a lot of different prediction tasks. But if you're thinking what we need in biology and what the ultimate goal is in biology, it's often quite different than what you would need or what you're thinking about in online advertising or recommender systems. Because in the biological field, what you really want to get to ultimately is something about mechanisms. I want to know if I want to study a particular disease and develop a particular drug. You want to know, which protein is misfolded and what actually happens downstream from that protein, and why, if this protein is misfolded, something else happens down the line, right? And so you need to really go from prediction all the way, or for many problems, you want to go from prediction all the way to causality or to mechanisms. Now, we of course know causality is hard, but you know, in the biological field, we have like these pretty substantial hammers that we can really perform interventions. Now, what are interventions in biological fields? So, for example, you can do things like, you know, knocking out a particular gene. So that just means setting the expression of that gene to 0. You can add a drug, like a chemical perturbation in the system, and see what happens. You can do mechanical perturbations and see what happens. And so if you're thinking about why are interventions so important for getting at causality, well, you know, let's talk about the recommender system. So in a recommender system, all we care about are really associations, right? I mean, Amazon doesn't make money by understanding that, say, if you're interested in buying sunscreen, that you may also be interested in buying ice cream. Right? They don't need to understand why that's the case. All you need to know is all the associations. That's more than enough to make money. But, you know, how would you now understand that, you know, that it's, for example, good weather that increases ice cream consumption, and that it's not the other way around, right? That causality goes this way. Well, you take like some cold day, rainy day, and you give out a lot of free ice cream, and you see that the weather doesn't change, right? That's the same, like you're going in and you're making an intervention downstream, and then something upstream doesn't change. That's how you're figuring out how causal relationships work. And so that's why interventions have so much power, right? If I intervene on a system, then whatever was upstream doesn't change, whatever is downstream may change. Okay, so interventions can really help you to get at causality. And that's what we have in the biological space. So we have the possibility to actually perform interventions. And but you know, the space of possible interventions is huge. So we still have quite little interventional data in order to learn all these causal relationships. But what I do want to talk about a lot in this talk is actually that what we do have is a lot of multimodality. So we do have a lot of different views on the same system. And we can perform interventions, and you know, we will be performing interventions in order to get at causality. But I want to talk about how multimodality can actually also help you just get at causality, even though it might not be interventional. Okay, and I'll give some intuition for why multimodality might actually help. Okay, so we do want to talk about causality, and we want to talk about representation learning, and in particular, how to actually intertwine the two things together. Now, why are current algorithms not sufficient? So here is a picture generated by DALL-E, and you know, just tells you, I mean, you know, all of the current methods, because there was always emphasis on prediction and not on figuring out like causal relationships or obtaining causal representations. Even if you tell DALL-E that it should draw you a picture of an ice cream stand where they're giving out free ice cream, it will always put good weather in the background. Okay, so even if you tell it was free ice cream, it will not make like whatever the distribution is, that if you would tell it like just draw me a park with people in it, it will sometimes have good weather and sometimes have bad weather, and you know, the probability will be not really 50-50, but it will maybe be like, you know, 30% like bad weather and 70% good weather. But if you ask it to do something with ice cream in it, it will always put good weather in the background, right? It cannot understand something about, you know, if I'm intervening in a system, what is actually upstream and what is downstream. And it was not built to figure out something about intervention, so it's not its fault. But that just tells you that, you know, current machine learning algorithms, they're just not built with thinking about causality or getting at causal representations. So we do need something different, right? And we do need to think a little bit more about it if you actually want representations that have something that can capture something causal. Okay, so what is the standard view of like how you represent causal relationships, or how do you think about regulation in biology? Well, you know, we have all these nice pictures of like ultimate goal would be, hey, I have, you know, all these different genes or these proteins, and then, you know, if I like, you have like some differential equations on this large network, and you would like to know that if I change this guy here and I toggle this particular gene down, then, you know, if I follow all these networks, it tells me that this one will go up and this one will go down, etc. This is kind of the ultimate view of like, if I would understand everything about regulation, then I could draw these huge networks, and I would really understand it fully. Now, how you would read these kinds of pictures is, you know, there is this causal framework that you can use to read these types of pictures that was already developed by Sewell Wright in the 1920s, where, you know, these directed causal edges, they mean literally something causal, that if I change this node, then actually everything downstream changes and what is upstream doesn't change, right? So this is known as causal structural equation models. And just to write it out, so you have like random variables, which are your nodes, which could be all of these genes. And every node is some function of its parents. So x2 is a function of its parents. So in this case, x1 and some noise, right? And so it also tells you what happens when you change a node. So if I set x2 to 0, then of course x4 changes, but everything upstream doesn't change. Okay, so these are causal structural equation models. They can model, also what happens under interventions. And now, of course, in biology, nobody tells me what the edges are, right? So the standard thing is you get measurements on the nodes, like how much of every gene is produced, and you would like to say something about these edges. So that's your standard causal kinds of questions. But very early on, so it's very nice work by Peter Spertus, who really laid the foundations of what can be learned just from observational data. But very early on, we realized that, so in humans, we have 20,000 of these nodes, right? So you would like to learn these networks on 20,000 genes. We saw very early on that these questions are just so hard from a sample perspective that you'll never be able to, with any technology, right now, you'll never be able to learn these networks on 20,000 nodes just directly from data. So even if you have, in fact, all kinds of interventional data, currently we get measurements of, like in every experiment, we get maybe 1 million cells, which is huge, of course. But even if you have 1 million cells, you can only learn a network on about 10 or 100 nodes. Everything else, we already know we'll get it wrong. Okay, and we'll get it very wrong. So it somehow just doesn't make any sense to even try because this scales really, really poorly. So we'll never get to learning a network on 20,000 nodes. Okay, we'll just simply never have the data set sizes to actually do something like this. So, of course, these are negative results, but I think it's kind of important to have these negative results because it tells you that, maybe this is not the right approach. Right? So even if you have all kinds of interventional data, like somehow learning these full underlying causal graphs just will always require a whole lot of prior knowledge. So then the question is, what can we do, right? So if this is, this is kind of what a biologist would always tell me, like that this is what they would like to have. But then, we would then ask, well, I can give you a network on 20,000 nodes. I know it's wrong, so how are you going to test it? Right? And generally, when you would ask that, it would be like, okay, so I'll make one intervention on some node that I care about, and then I'll see whether you get the downstream effects right. Well, then let me just do that instead of trying to learn a full network, which I already know I'll get wrong. Okay, so then, let's try to think about other questions that are actually relevant problems that may be easier from a causal perspective. And so here I'll put like 2 types of questions that we have been working on for a while now, which I think, and capture like a whole lot of questions that you're interested in the biomedical sciences. So what are these questions? So here's the first one. So say you have all kinds of different interventions. These interventions can be drugs that you apply to a cell, or it can be these genetic perturbations, so I knock out a gene or whatever. Any kind of interventions that you can do on cells. And I get to measure some output, okay, gene expression, an image, whatever you want to measure. And so now say you have this huge drug screen or this genetic screen performed in one context. The context can be, I have it performed on skin cells, and I would like to then know what happens for the same perturbations on a neuron, for example. Or I have it performed, there are large-scale data sets in the cancer setting. They have done like a whole lot of drug screens and genetic perturbations on cancer cells, different types of cancer cells. Now comes a new disease like COVID-19. Can you, without redoing the whole drug screen and the whole genetic screen, can you tell me what happens when I apply the same drug in that setting? Okay, so that's a context switch. Or ultimately, what you would love to do is, you have all these perturbations applied in the mouse. Can you now tell me in the human what will happen, right, when I'm doing that? That's the ultimate kind of context switch that you would like to be able to understand. So this is a transport between different contexts, okay, which underlies a whole lot of questions that you might be interested in. Here is another one. Equally important, I think. So we know the space of all drug-like molecules is something like 10 to the 60. So even if you know which disease context you care about, you'll never be able to do all 10 to the 60 many experiments. Or, if I'm looking at these genetic perturbations, I told you we have 20,000 genes, but we care about all combinations of genetic perturbations. We'll never be able to do them, not just two-way interactions, but three-way combinations, et cetera. We'll never be able to do them. So the question is, even if I already know which context I care about, say a particular cell type in a particular disease context, and I have like a large-scale perturbation screen done on them, well, I would like to be able to predict what happens under other perturbations, right? So another drug that I haven't seen, or another combination of genetic perturbations that I haven't seen yet. Okay, so this is a different problem. This is transporting to new perturbations or interventions. Okay, so you can transport to new contexts, or you can transport to new perturbations. So these are the types of questions that I'll talk about. Now, of course, if I know the full underlying causal graph, I can answer these questions. Okay, if I know the full underlying causal graph, like what I had before, and you ask me, okay, I know what happens under this perturbation, can you tell me what happens here? I can give it to you if I have this understanding. But as I said, there is no way you can get this understanding in all of the biological contexts. So maybe this is an easier question, okay? So let's just, because these are actually the questions that will be, that's how a biologist will try to test me whether I get any causal reasoning right, because this is what they will care about in the end. So let's maybe just try to go after these questions without going through the whole trying to learn the full underlying causal system. Okay, so already how I wrote it out here, it's clear that this is a generative question, right? Because I'm asking you to generate the data that corresponds to applying this chemical perturbation in this new disease context. Okay, so let's think about, and I'll just present the types of generative models that we've been using for these types of questions. So I'll take a little bit of a detour to motivate the types of generative models that we've been using, then get back to this question. Okay, so here are two of the, I find, some of the most exciting works in machine learning from the recent years, or that have made us rethink how, what neural networks are actually successful in many different types of applications. So Misha is just giving the talk now at the same time, and the Neyman seminar. So we have a lot of joint work, and you'll see a lot of joint work coming up now. So he has had, with co-authors, has had this work on, we all have seen that neural networks have become very successful once they were large enough. In fact, had many more parameters than the number of samples, right? So they can all be trained to zero training errors. And the question was, how can they generalize, although they're trained to zero training error? And so he had this work where he shows that you can still generalize, although you're interpolating the training data. Then if you take this to the extreme, you can say okay, so there is motivation for making neural networks larger and larger. Now, of course, generally larger and larger came with a computational cost. But if you go all the way to the limit by making it larger and larger and letting actually the size of the width in this case go to infinity, you actually suddenly get to the setting where you're not paying a computational cost anymore, because that converges to a kernel, which we know is this neural tangent kernel, which you can write down in closed form. Okay, so suddenly you don't actually have to train anymore. So it's this kind of dichotomy, right, where, yes, you get better and better models, you make them larger and larger, you pay a computational price, but in the limit you don't. Okay, so this is all in the classification setting. And, something that I actually found kind of surprising when we're working on this is that although these neural networks are used in all these, pretty mission-critical domains, there was actually never even a proof that they're optimal. Even in the classification setting, the simplest classification setting, that they can be Bayes optimal. We know neural networks can represent any function, but the question is, if you train them, are they actually giving rise to a Bayes optimal classifier? And so we recently proved this. This is, again, work by my PhD student, Adit, and you'll see him come up multiple times together with Misha Belkin. And we showed here that now let's take these infinitely wide neural networks, and let's make them also infinitely deep. So that, we have all that matters. Now the architecture is out of the question. I made it infinitely wide, infinitely deep. Now all there is to toggle around is the activation function. Now it's nice, right? Nice math. And at least in this case, we can actually show that neural networks they can be base optimal, and it now only matters on how you choose the activation function. But the optimal neural network is not your standard ReLU or sigmoid or whatever activation functions that people have been using, but it's actually not a hard one. Okay, so some cubic polynomial as an activation function is in fact optimal. So this is, this is in fact a base optimal neural network, but it is new types of activation functions that one should be using. Okay, now this still doesn't tell us that your standard neural networks, that are not infinitely wide and deep, can also be base optimal. We simply don't know. But at least this motivates that we should be looking at these very, very large neural networks. Okay, because in this case, at least we can prove that they are the optimal thing you could possibly do. So this is still old classification settings. So now we care about representation learning, right? We care about these generative models. So then still this kind of suggests, so now we have to ask, what does overparameterization mean in the representation learning setting? So we use autoencoders just because by now we have a pretty good understanding of what these models actually learn. And so this is your standard autoencoder, right, with like a neural network used to go to this latent space, which is standardly a bottleneck, and then back to your original space. Now, overparameterization in an autoencoder will mean the following, that you start in some dimension, say 20,000 for gene expression data, and we're actually going to map it into a larger dimensional latent space. Okay, so instead of your standard view of autoencoders as dimension reduction, we're not going to do dimension reduction. We're going to map it into a larger dimensional latent space, and then of course back to your standard space. Now, nobody has done this because it sounds crazy, right? Because now, nothing keeps me from not just learning the identity map. But that was kind of the same here, right? That's why, nobody thought that you could use these overparameterized neural networks because, if they just interpolate the data, then maybe it's not a useful function. So here, again, it's the same kind of thing. So there are infinitely many functions, like here. There are infinitely many functions that interpolate the data. Some are good and some are not so good. Okay, the identity map would be a pretty bad map because I wouldn't have to do anything, right? If this was just the same representation as out here. And there are papers that suggest that these autoencoders learn the identity map, which is wrong, because I can easily, and we have a proof, but you can just do it yourself, like, try to train such an autoencoder. If it was the identity map, yes, whatever comes out, it looks kind of like the identity map. If it was the identity map, whatever comes out, you put it back in. I can iterate the map. If it was the identity map, nothing changes. Okay, but if you do it, you will see, and we now have a proof, that whenever you iterate, so these overparameterized autoencoders, the functions they learn are attractive at the training examples. So if you iterate the map many, many times, so this is already it. So here we just take a training example, we put on top a whole lot of noise, okay? And now, this is now a test image. You put it into your autoencoder, you see what comes out, and you just iterate the map. And we have a proof, but here you see what happens. You will always converge to one of your training examples. Okay, so definitely doesn't learn the identity map. Now, of course, you can ask me, why is this a good thing? Now, the nice thing is, like, people like Benjo, very early on, when they had like autoencoders, they actually added another loss term to make them attractive at the training examples. Okay, I'll explain why that's a good thing, but they already understood very early on that this is what you would want your maps to be. And now what this says is like, just over-parameterize and you get that for free. Okay, so just minimize reconstruction error, make them over-parameterize, and you will get your behavior just for free. You don't have to add all kinds of complicated loss terms. But why is it a good thing? Well, if you have your training example somewhere, right? If I give you a test image or a test example that is close to a training image, you definitely don't want that to be mapped to something very far away, because you're pretty sure about how the manifold looks like close to your training images. Right, this is what this says. It says like, okay, if I give you something kind of close by, well, you will be mapped a little bit closer to your training image, but minimally closer. I can, like when you're putting it back in, when you're putting a test image in, it looks kind of similar, but at least, you will be mapped to something closer. And then if you iterate many, many times, yes, you will converge to one of the training images. Okay, this is a good thing. You definitely don't want to be mapped to something very far away, right? Close to your training images. Now, of course, there's no standard definition of what does generalization mean for representation learning, but this would be our suggestion of what you would want it to be is somehow close to the identity map, but still attractive at the training examples. Okay, so these are always the autoencoders or the frameworks for generative models that we're going to use. Okay, these autoencoders that are highly overparameterized, that have these large latent spaces that we're going to use for these types of questions that we have here. Okay, so that was a little bit of a detour. Now, next question of getting to causality, okay? So I started out by saying like how we generally think about these causal systems, right? It's always these large networks where I have the nodes be, say, genes, and then the premise of causal inference is always I give you the nodes, I give you data on the nodes, you have to learn the edges. Well, okay, so now I give you these, that's why I have here another example. Data can often be images, and images are much cheaper than gene expression data. So if I give you images as an output, well, what are even the causal variables? Okay, so pixel 1 is certainly not a causal variable because pixel 1 has nothing to do with pixel 1 in another image. Right? I mean, the cells have all kinds of different shapes, et cetera. We don't know what the rotation is, et cetera. There's no way to just orient them to make pixel 1 map to pixel 1. It's not like before where we had the networks where, I mean, we think we know, or like just because of like how these, how these gene expression vectors were measured, we just put the node for every gene and then built these like regulatory networks. But it's not clear, that was maybe misleading because it's not clear that genes are actual causal units. It might be combinations of genes that should be a causal unit because they always work together. And this other combination of genes that should be another causal unit. So it's maybe misleading, but it's very clear in an image that we don't know what the causal units are. Right? We first have to learn what the causal units are. The causal unit might be like the shape of the nucleus. It might be how much of a particular protein is inside the nucleus or where it is located, et cetera. We don't know it. We have to learn it first. Okay, so really, in biology, the problem is much harder, right? It's like we first have to identify what are even the nodes, and then we have to learn what are all the regulatory relationships. Okay, so that's why it's causal representation learning. It's really learning a latent graph so learning first the latent nodes and then learning a causal graph among the latent nodes. So that's really the problem that we're dealing with in the biological system. Okay, so now the question is, how do we learn these latent nodes, right? How do we even get to what are these causal variables in this biological system? And it's very, very clear if you deal with images that you have to learn what the causal variables are. And so here is the suggestion, and I think intuitively will make a lot of sense, and we finally have some proofs as well, that maybe having data from multiple modalities, even if it's not perturbations, might help you get a causality. And so here I first just have a little bit of intuition for why that's the case. So what is hard about causality? Well, what is hard about causality is that we have all these associations that we see in data, and we need to figure out which associations actually come from direct causal relationships, and which ones just come from long paths, or because you have some confounders, or because you have like something that you're conditioning on and it's opening up some paths. That's what is hard about causality. It's about throwing out associations that you see. It's not about learning new things, right? It's always about you see a whole lot of associations. You need to throw out the ones that are not causal. That's really the hard part. So here is just some intuition, right? So whatever is causal should be there in the system, whether you look at it from the right or from the left or from the top or from the bottom. Okay, it should be somehow invariant to how you're measuring it. Whatever is causal in the cell should be there, right? No matter how you're measuring it. So that's the intuition how multimodality might help you, right? So if I'm looking at a cell from imaging land or from sequencing land or from whatever land, there will be always some biases and some associations that you're building in just because of the measurement process. So if I have many different ways of looking at a cell, then maybe what is invariant to how I look at the cell is much more likely to be causal. It might still have some associations that I haven't been able to throw out that are still not really causal, but actually having many different views of a cell might help you to distill whatever is causal. Okay, so that's just the intuition for us to build always these multimodal. So this is now an autoencoder that goes from one modality to a latent space and then back. And you have other modalities and you build them into the same latent space. Okay, and you just match them in the latent space. If you have paired data, you can just match them so that, if you have a cell over here and a cell over here, they're the same cell, just viewed from different points. You just have a loss function to make them match in the latent space. And that also then allows you to kind of translate between different modalities. We did it for heart ECG and MRI data so that you can now translate between many different modalities. But that was the intuition. It's always like, maybe, whenever having many different views of the same system can actually help you get at some causal or some, throw out stuff that is kind of not important just because it is not represented. It is not invariant to how you're actually looking at the system. Okay, that's at least the intuition. Now we finally have a proof of something like this, and let me show you what are the kinds of things that we can prove. So as I said, so causal representation learning, so we have a causal graph, but the causal graph is among completely latent variables. Okay, so every variable here is latent, right? So these would be my programs, these biological programs that I don't know yet. I don't know whether the protein is inside the cell nucleus or what is the shape of the nucleus, etc. And now you have different functions that map you from this latent space to the observed space. This could be sequencing, this could be imaging, this could be whatever other modality you have. Okay, now you didn't stop me before on the previous slide, but probably some of you have been thinking exactly this, that, in some modalities, there should be some causal variables that I can only measure in one modality, which I will miss in the other. Okay, so before I was making it very easy, I was saying like, okay, whatever is not there in any of the, is there only in one modality shouldn't be causal. That's like trying to throw out as much as possible to be very, very sure that I get something causal. But you could have some latent variable that I can only measure in this one, right? And I can have another causal variable that maybe I only see in imaging land, but I'm not able to tell it apart in the sequencing land. So that's what we're allowing in this theoretical framework. Okay, you see some variables here where, this modality here is a function of these shared guys plus one modality-specific one. And now, so the question is, if I just give you data here from many different modalities, can you actually identify these latent variables? So the first question is even how many latent, we don't even know how, right? Because it's all latent, I don't even know how many causal variables I have. And then there is a question of what are they, and then there is the question of what is the causal graph. And so, okay, so here are some, of course, you start when you want to do something like this with very simplifying assumptions. So at least if everything is linear, so we have, linear effect here, linear causal effect. I'll talk about non-Gaussianity in a minute, and then you have like linear functions down here, then in fact we do have this theorem now finally that that it's the kind of intuition is right, that you do get identifiability just from having multiple views on the whole system. Okay, so by having these multiple views on the system, you do get to identify what actually the causal variables are. I need to talk a little bit about non-Gaussianity. It becomes very clear if I go back. Let's do the following here, where we had, we take a population of cells, we take out some for imaging and some for sequencing. Okay, and in fact, so in the problem afterwards, so in these kinds of two modalities, both the modalities are actually destructive to the cell. So if I get a cell, I will never be able to see it in imaging land and in sequencing land. Okay, so many of the biological measurements are actually destructive in that way. So I never have these paired measurements in here. Now that's hard, right? So the question is, how do I match the latent spaces to make sure that, because for this cell, I'll never be able to see what it looks like in expression land. So then the question is, how do I align the two latent spaces? Now, the way how we did it here, and it will be clear why we need non-Gaussianity, is, if I take a population of, say, I don't know, immune cells or something, right? And I take out at random some for imaging and at random some for sequencing, then I know that it's the same population of cells, so the distribution has to be the same. So I will just, in the latent space, make sure that the two distributions align. How you do that is you can, for example, add another classifier that tells me, if you can tell, I give you a cell in the latent space, if you can tell me whether you come from imaging land or sequencing land, I'm doing a poor job. Okay, so I'll be penalized if you can somehow figure out whether I come from sequencing land or imaging land. Now, of course, if you have a Gaussian distribution with all of its, all of its symmetries, well, there are many ways to align two Gaussian distributions, and there is no way for me to tell which one is right. Okay, so that's where, which already tells you from the application why, in the theoretical results, you will need like non-Gaussianity here in the noise. Okay, but non-Gaussian, generally your distributions are not Gaussian. In particular, if you have many different cell types, et cetera, then you already know that the distribution is not Gaussian. Okay, so just, somehow all of these assumptions, or some of these assumptions are very, very, intuitive. You already know that you will need it. Of course, linearity is not something that it's definitely something that we would like to get rid of. But, these results don't tell me that in the nonlinear setting it's not true. It's just that if you have linearity, at least we're able to prove it. Okay. So the intuition at least goes through. Now, you can also think about, this was kind of nice because we were asking just multimodality, right? How much can we get at causality? But of course, if we have interventions, we should be able to do at least this, right? These results will go through if like this modality is intervention on node 1, this modality is intervention on, whatever, add another chemical, and this modality is knock out gene 3, the same result will go through, right? These are different modalities. But maybe I can do more. And so the question is, if I have actually perturbational data, can you do more? And in fact, we can prove these same results, but now we don't need any of the simplifying assumptions. Okay, so fully non-parametric, so, completely non-parametric, no assumptions on any of what the distributions are, fully, non-linear here, so completely non-parametric, and all of these functions can be polynomial. Okay, so I have that on the next slide. So in this case, when actually every one of the different modalities is an intervention, then we get like a very nice general theorem on like everything non-parametric here in the latent graph, and then this function here being polynomial. Okay, so that's how much more you can prove. We don't know that the previous cases that we couldn't also get similar results, but at least in this case, we can actually prove it when you have interventional data. And even more, we can give you an algorithm that will identify the latent causal graph when you have interventions. And so what is the algorithm? So it's this, again, an autoencoder. And okay, so how does this work? Okay, so you have here it's only one part of the autoencoder, or I guess kind of both parts. But so what am I going to put in? I'm going to put in observational data, so without any intervention applied to it. Okay, so say I give you an image where no drug was applied. And what I'm trying to decode for every intervention is I'm trying to generate data of what happens when I actually apply this drug. Okay, so I'm not just using an autoencoder standardly where you try to minimize reconstruction error, but now I'm putting in observational data, I'm giving you which drug it is, and then you should generate the corresponding images under that intervention. Okay, now we have a problem that, of course, with interventions, I either see the cell with an intervention or without, I never get to see both. So I don't know if you give me one cell how it will look like here. Right, because I never get to see both. So that's where this, this distributional loss has to come in, or this max mean discrepancy. There are other kinds of distributional losses that you can take, but you have to do some batching, right? I give you a whole batch of cells, observational cells. I generate a whole batch of interventional cells, and I have like a loss on, the whole batch of cells together. Okay, you need that, right? Again, like before, I need to match distributions. I cannot match at the single cell level because I don't know how they're paired. Because you never get to see both examples at the same time. And where does causality come in? So you have here a causal graph in this neural network. So you have here a nonlinear function that maps you to this causal graph that you're actually learning. And then the causal graph is exactly this causal graph among the latents. Okay, so we have proofs on that. In fact, this kind of architecture will give you, if you have at least one intervention on every one of your latent nodes, it will actually guarantee you that you're learning the correct causal graph among these latent nodes. So it's quite nice that we also have the algorithm and an algorithm that is completely nonlinear, et cetera, where you can prove that you are really getting identifiability of this causal graph. Okay, so. I will have 5 more minutes, right, where I should stop. So I still haven't completed this question of, so I'll just finish with, how are we actually going about really now figuring out this problem? So what I did until now is okay, if you, so I give you some interventions, right? Can you then generate what the effect will be of other interventions? That's kind of what the previous problem can do, or the previous formulation can do. But now let's ask about this, right, generally. If you would like to, from some given perturbations in one context, can you tell me what happens for these perturbations in another context? Or very generally, if I have different perturbations, can you generalize to other types of perturbations that you haven't seen? Now, there is actually very nice theoretical work by Barenboim and Pearl, which says the following, that if you give me, and I already told you I cannot, but if you give me the full underlying causal graph, okay, and you tell me that this perturbation corresponds to this node, and this other perturbation here that you care about corresponds to a perturbation on this node, okay, so you tell me everything, the full underlying causal structure, you tell me where the perturbation is, then they have really strong if and only if conditions on whether there exists a formula, and they also have the formula, that you can tell me what, if I know what happens when you intervene here, they can tell you what will happen when you intervene on this node. Okay, that's fully characterized, so necessary and sufficient conditions. This is known as causal transportability. So, I give you one intervention, you tell me what happens in another node. But as I said, we don't know the causal graph, right? But these are the types of results that we would like to get. It's this is the problem we want to solve. So if you give me the causal graph, they have it completely characterized. But of course, we don't know the causal graph. Okay, so here's how we're thinking about this problem in a very, very simplified manner, and it will kind of tie everything together that we've done until now. So this is a particular data set. I talked about the cancer data set. So these are all different cancer cell lines. So say, I don't know, one is some skin cancer, and then another one is a lung cancer, et cetera. So these are many different cancer cell lines. These are many different perturbations. Here we're only taking, in this particular application, we only took FDA-approved drugs, but you can take, they have thousands of different perturbations in this data set. It's all freely available, this data from the Broad. Every dot here means that gene expression, so in this case, only a 1,000-dimensional vector was measured. Okay, so this drug was applied to this cancer cell line, and you know what happens when you do it. So you see, for example, the data is very, definitely non-randomly distributed. So everything white means this experiment has not been performed. Okay, this is a pretty expensive experiment, so you'll not be able to fill in everything. So this is the transport question that I had on the previous slide, right? One is, you have seen this particular drug in this cell line. Can you tell me what that drug will do in a different cancer? Or the other way around, in this particular cancer, I know what this drug does. Can you tell me what a different drug does in this particular cancer? Okay, these are the two transport questions that I had. Now here it's pretty clear you can view it as some tensor completion kinds of question. Now, any of your standard completion approaches always is based on whatever it is, it will do something based on low rank behind the scene. Now, low rank will do very, very poorly, right, if your data looks like this. So just think of the extreme when I only have one observation here. Like, for example, COVID, a new disease comes along. Very quickly, we knew how the expression profile looks like in an infected cell. well, but none of the drugs were applied to it. Well, if I do low rank, I will just plug in the constant everywhere. Okay, that's a pretty bad prediction for, so any of the kind of your standard matrix completion algorithms will not work. Well, so what you can do, and it's actually not so hard to do, is now I talked about neural tangent kernel, right? And all the motivations for why you should maybe be looking at these types of functions that are just neural networks that are infinitely wide and possibly even infinitely deep. And you just write out, this is math, etc. You write out what the neural tangent kernel means for matrix completion. Okay, so you try to figure out what it is, a nice closed form solution, etc. And all we did here is just applied something principled, like neural tangent kernel for matrix completion. And you can just by simply doing that, you already beat many of the methods that were developed for this type of question. Okay, and of course this is super fast, right? Because I don't need to train anything. I'm literally just plugging in this data for matrix completion using a neural tangent kernel. So that's, and it doesn't do low rank. Okay, so what it uses, because it's a kernel, you can also figure out what does it actually care about, and it's pretty nice. So you can use, it has a place where you can put in priors, and so what it will use is, it does inner products, of course, because it's a kernel. So what it does inner product, or what the function is, the inner product is among, it looks at all of the other cell lines, and it looks at all of the other drugs, and it does inner products between two drugs and inner products between two different cell lines. And so if the inner product is large, it makes use of cell lines that look more similar to itself, and it makes use of drugs that look more similar to itself in order to figure out what it should fill in. So it does something very, very intuitive, and you can figure it out because it's a kernel. You can actually figure out what is it in fact using in all of its arguments. Okay, so that's one way of how you can do these causal completion problems without actually doing something that is just simply low rank. Okay, so where are we heading? Of course, in the end, you want to get to control. So just being able to predict every type of intervention is not the end, right? You would like to give me a healthy cell and a disease cell, and you would like to know which interventions to do in order to get there. Or what we care about a lot is, changing the type of a cell. So I give you a skin cell, and you would like to make a neuron out of it. What are the interventions you need in order to get from one cell type to another? So these are the types of things we do now. I won't go through it, but I think there is a really unique opportunity to change, and I know many people here are excited about experimental design in the protein space, to really change how experiments are done, right? If you're thinking about it from a more principled perspective, you have a particular state you would want to get to, and you have a lot of rounds of experiments. So we should do that in a principled manner and not just like hill climbing. There is a little bit of exploration and then exploitation to be done, and this can be done in a principled way. So I think that's a really exciting framework. Okay, so I hope I was able to convince you that, the biomedical sciences are not only uniquely suited to just using machine learning that has been produced, motivated by other types of applications, but actually also is one of the greatest sources for inspirations of new foundational developments in machine learning. I talked about over-parameterization. I think there is still a lot of open problems about how to optimally combine representation learning with causality. And I think I just want to end with this Julia Pearl kind of setting of like, he has like these three, the ladder of causality and the three different settings. So you have down here associations, which is, your standard where neural networks are applied to, which I had like this application in this recommender system of, just understanding whether if you buy sunscreen, you might also be interested in ice cream. You have counterfactuals, which is what all of causality has actually concentrated on, asking questions like, what would have happened had I not taken the aspirin? But there is actually this level in the middle, which he couldn't care less about. This is where Neanderthals are. But this is the level where you ask questions like, what will happen if I take the aspirin? And in biology, I would argue that, in the medical sciences, you do care about counterfactuals in clinical trials, et cetera. But in the biological setting, I don't care about things that we cannot validate. We will be performing the experiment, right? What I want to do is some drug screens, virtual drug screens, but in the end, you will be performing the experiment. So I only care about questions where I'm asking, what will happen if I intervene in the system in this way? And so all of the talk was actually at this level here, and that's why maybe we don't need to go to all these causal models of like learning the full underlying causal structural equation model, et cetera. That can answer questions that are counterfactual. So maybe we can take some shortcuts because we're actually staying at this level. But this is really the level that we care about a lot in biology. So if we can just come up with a full theory of how to answer these types of intervention questions, I think we'll already be able to do a whole lot. Okay, with this, let me end. This work was really all performed by amazing PhD students. So Adit Radhakrishnan did all of the over-parameterization of neural networks. And then Chandler did all of the causal structure discovery, so when you're actually learning these causal graphs. Jachi did all the work on causal representation learning that I presented, and she needed all of the biological applications of integrating and translating between different biological modalities, et cetera. And of course, this was in collaboration with a lot of different amazing people. I talked about Misha Belkin's work. All of the biological work was Shiva's lab, and I had multiple other papers that I talked about. And of course, without funding, wouldn't be possible. And thank you all for your attention. And happy to take any questions.
This is the mic that gets thrown. Any questions? I can throw it really hard. Okay, I'm going to start off with some questions. So at some point you said that different views gives identifiability, and was that like a kind of precise statement? Or is that just the kind of rough statement of if you have one observation per intervention per latent node, then, or there's two separate things?
Yeah, so this is the precise statement. But before we had the imprecise statement, which actually worked, but then we did the precise statement. So this means you have like all of your latent, right? All nodes are latent, and you have your causal graph among the latents. And then every view is I have one function of my latents, and I have another function. This would be the measurement process. So I measure from these cells, I measure RNA-seq. And here I measure images, and here I measure something else.
I guess I was wondering, like, how different do those, what if it was each of those were the image, all the same thing or something? Like,
Perfect. So then I would have basically nothing in the outside. Then it's, right, then I don't, so if everything is the same, then I wouldn't have any modality-specific latent nodes. So we did this also for example, one thing is batch effect removal. Right, so there you only care about what is, so there already it's pretty clear, right? So if I have different batches, it's pretty clear that whatever is causal is certainly not, is only what is invariant to the different batches. So that's the intuition here. This is just more extreme because like two different views are literally like the kind of associations that are not causal will be pretty orthogonal. And so that's why it helps you even more if you have very different views about the underlying system.
If that came into your theory, like how different those views are and quantifying that. And
it comes in, yes, and we don't have all the theory. So here we're just saying in the theory you say it's very different. But you do see how having many different views helps. So that we already have in here, but like really quantifying everything. Yeah, so the intuition at least, it comes through, but we haven't quantified everything carefully. Yeah, we also have infinite data, et cetera, and all these results. So obviously, yeah.
Oh, we got one. I can try it. Ready?
Uh-oh. Woo.
In your polynomial causal thing, you said that you have to record intervention at every node.
Oh, yeah.
How do you make sure
very good.
experiments at every node is observed?
Yes, that's another assumption that I don't like, but it makes sense in the genetic setting. So in the genetic setting, we now have whole genome perturbation. So we have a perturbation on every node. So then what this result tells you is actually something interesting. What it does is it maps every intervention to one of the latent nodes. Okay, so in some sense, so then I'm learning gene programs because now it says, it also gives you interpretability of what the latent node is. It tells me this latent node corresponds to gene 1, 3, and 5 because all the interventions were mapped here. So that's what we will be calling a program. For images and stuff and chemical perturbations, this assumption makes no sense, right? Because how do I know for a chemical has multiple, has multiple targets, etc. So we would need complete. So for chemical perturbations, all we have for now is just this result, right? It cannot tell you how chemical perturbations are better than just seeing it as a different modality. So applying chemical one means this the modality, and then the other one means this modality. So the next results really only work for genetic perturbations.
For chemical drugs, is there something that you can say there's actually a latent variable missing? That your graph is incomplete on a new drug, or?
So, so these results will always only tell you what are the latent variables that are shared among the different modalities. Now, if I wouldn't have had this modality, I would never have gotten to CX5 because this is modality specific. So it doesn't tell me, it just learns what are the latent variables in the causal system that I currently get to see. And those are really great questions, right? So that would be awesome if we could say like what is missing and what should we still be measuring. But that's not what you get out of this. Yeah.
Yeah, I have a question. It is about you mentioned the identifiability. If I understand correct, you mean the reconstruct the causal graph perfectly. So I'm wondering what.
Can I just say it's actually so more than the causal graph? I actually care more about what are the latent variables. So identifiability first even means to tell me how many latent nodes, how many latent variables do you have? And then what are the latent variables? And then, a second part is about the causal graph, but I actually, I think the more important problem is to identify what are the causal variables, not so much the regulation between them.
Yeah, the question I want to ask is, what is the identifiability, in what context? Is there up to some equivalence class or something like that?
No. No equivalence class. So before, when you have fully observational, you often have equivalence class. But in fact, you can, under non-Gaussianity, you can fully identify the causal graph. So here again, even if you look at the causal graph, it's fully identifiable by if you care about the second part of the causal graph stuff and whether you can learn it, it's always the full causal graph.
Yeah.
But that just comes out of non-Gaussianity. Yeah. But I think the first part is more important. What are the causal variables?
Maybe this was answered and I lost it, but once you have the number of latent variables, even just finding the ordering, finding the structure is sort of an NP hard problem, right? Even if you know those units, and so I'm just
correct, that's
computationally,
correct.
is this a
That's
search
correct.
process or?
Yes, so how you would implement it, so this, okay, so in this problem here, this is a theoretical result of that it is identifiable, and we have some algorithm in there that I would never apply in actual real setting. Now here, if we have this interventional data, I have an algorithm, right? But again, actually figuring out what, so why this is not NP hard, why it can be solved with this algorithm is the following, because here you just start with an ordering, which is a DAG. Now, what it does is it maps every one of my perturbations, right? Every gene perturbation will be mapped to one node. I can just fix the ordering. All it matters is where do I assign the variable.
Okay.
So it's just an assignment.
And then the MMD, it needs a kernel, does it? Does MMD require a kernel choice?
So yeah, so we didn't optimize on any of these types of things.
Okay.
Yeah.
And did you have any thoughts on the importance of that, or?
I am sure it will matter. I'm very sure. Yeah. But yeah, still a lot of many more problems than answers.
Very cool work. I'll save more of my questions because we're already over time. Any last? We got one.
So I had a question, so whenever you go into the latent space, I'm not sure if this is something that you care about, but is there a way to get out of the latent space such that you can identify at least for the picture example, if you go back out of the latent space, can you identify like which part of the picture is relevant in terms of the causal relationship?
Yes. So I didn't talk about it, but obviously now that we have these results, we would like to apply them, right? So I would want to, and we have. So this would be images, and this would be, say, sequence information. So I would like to understand what are the causal variables that are shared. So what is information between two very different modalities that is actually shared between them? And what are these variables, right, that is just modality specific, that I can only see in an image or that I can only see in sequence? And so that's exactly what we did. So yes, you can go from here to actually then understanding what is in here and what is in here. And so how you do it, and maybe for now it's hacks, but like, so you have a latent space, right? So I have, like, for every cell, I have a representation in like some, like whatever. Now in this case, I would have 7-dimensional space, okay? So now you look at all of these dimensions here. I just only took out, okay, let me do it in 2 dimensions. So I take out these 2 dimensions for all cells. I do PCA. Okay, so now so let me take just the first principal component. So every cell will land somewhere along the first principal component. So I just take the most extreme ones. And now these are in the latent space, so you can map them to your observed space, right? And now I just look at how are the 2 extremes different. Because it's autoencoders, I can always generate the pictures corresponding to it. And so then we do see things like that come out, like we did it for a TAC-seq and RNA-seq, for example. So you get like things that we already know, like, RNA-seq tells you something about cell cycle, a TAC-seq doesn't, et cetera. And for images as well, we found things that we can, that is only contained in chromatin images and is not contained in proteins. But that's how you can do it with autoencoders.
Okay.
Yeah. But we don't have a principled way, but this is for now how we're actually really being able to identify what are these variables and what are these variables. Yeah.
Right. Well, let's thank Caroline once more.
Yeah. Thank you very much.